Hello sir, this is Kathleen Rose of Labog from BSEd. This is my laboratory 3. The process of transcription and translation. What is transcription? Transcription takes place in the nucleus. It uses DNA as a template to make an RNA molecule. During transcription, a strand of mRNA is made that is complementary to a strand of DNA. Transcription is the first of several steps of DNA-based gene expression in which a particular segment of DNA is copied into RNA by the enzyme RNA polymerase. Transcription takes place in 3 steps: initiation, elongation, and termination. The first is initiation. It's the beginning of transcription. It occurs when the enzyme RNA polymerase binds to a region of a gene called the promoter. This signals the DNA to unwind so the enzyme can read the bases in one of the DNA strands. The enzyme is now ready to make a strand of mRNA with a complementary sequence of bases. The second is elongation. It's the addition of nucleotides to the mRNA strand. RNA polymerase reads the unwound DNA strand and builds the mRNA molecule. Using complementary base pairs, there is a brief time during this process when the newly formed RNA is bound to the unwound DNA. During this process, an adenine in the DNA binds to an uracil in the RNA. The third is termination. Termination is the ending of transcription and occurs when RNA polymerase crosses a stop sequence in the gene. The mRNA strand is complete and it detached from DNA. The last is, what is translation? Translation is the process by which a protein is synthesized from the information contained in a molecule of messenger RNA. During translation, an mRNA sequence is read using the genetic code, which is a set of rules that defines how an mRNA sequence is to be translated into the 20-letter code of amino acids, which are the building blocks of proteins. The genetic code is a set of 3-letter combination of nucleotides, called codons, each of which corresponds with a specific amino acid or stop signal. Translation occurs in a structure called the ribosome, which is a factory for the synthesis of proteins. The ribosome has a small and large subunit and is a complex molecule, composed of several ribosomal RNA molecules and a number of proteins. That's all. Thank you.